Wow. Hello, everybody. Hello and welcome. That was the first time that we had a clap on like this. Thanks for the person here initiating this. No, welcome to the already third event of BluBulB that we're doing. On the first time we had an event roughly 2 months ago here, I was quite nervous because hopping in front of a new crowd is a bit like, okay, what you're gonna meet, who are the people there, and it was nice. On our second event, I was just excited to hop in here now, and I knew people are nice and we have something good, so it was pure excitement. And now I'm just simply happy to look in so many faces, expecting faces, which is also really not good. Thank you all for coming, and a special shout out to all the people that were here on the last event or even on 2 of the last events. We're really grateful that you're coming again and show that actually what we're doing is somehow interesting, nice. We are a young initiative, BluBulB. That's only our 3rd event. That means we still want feedback, we still need feedback, and on your tables you should find some small clip outs with some QR codes. If at some time during the event today you will take the time, scan this QR code, go there. We want to know what you think about this, what do we do good, what do we don't do so good yet, and who are you? We want to know our audience, every single one of you. Also, I should mention we are filming here tonight. That's just a legal thing that I need to mention this. Or also, if somebody of you is here on a secret mission by Her Majesty or by somebody else, let us know and we'll figure out what to do about this. Before we get started, let me quickly tell you what you got yourself into. BluBulB is an initiative that's funded by CPR, the Center for Protein Research, here at the Copenhagen University. And we are 3 colleagues, and we want to bring back this fascination, this wow of finding out something new, being curious about something that we all had once when we are younger, and still many people do. So we invited 2 scientists tonight, and they're going to use 15 minutes, not only to show what they are working on, but also to show why it's cool and why it's relevant and why it's interesting. On top of this, because science can get a bit hard sometimes, though we make an effort to make it as easy as possible, we also have an artist here tonight, and he will give us a different viewpoint on a topic that we are looking into tonight. We also have Eleonora here tonight, who is sitting over there, and she will sketch along. She's listening just as you do, as I do, and will draw along to the things that she's hearing. And we'll check in regularly with her to find out if the presenters, the audience, and Eleonora, if we all are getting the same idea, the same concepts out of this. It is also Eleonora that we sent on small reportage of a couple of minutes to our contributors tonight to find out where are they working, what are they working with, and what do they do. So with all of this being said, we have a topic for tonight, and this topic is communication. Communication, in its most simple way, is just the transfer of information from a sender to a receiver, and in between there is some kind of message. This message can be 2 molecules that tell each other what to do or not to do. This can be 2 people that have a hard time understanding each other, or that can be signals that are coming from deep space or from a long time ago. But I'm already getting ahead of myself. We're going to start with a biologist. She is working with proteins. Her name is Charu, and we're first going to meet her in a small video, and after this, here on stage. Have fun, everybody.
We communicate all the time, but have you ever wondered how protein in our cells talks with each other? We are here to figure it out with Charu. She's a Ph.D. student at the Novo Nordisk Foundation Center for Protein Research. Let's do that. Ciao, Charu. I'm fine. You?
I'm good, I'm good.
Are you bringing us where you work?
Yes, let's go and see around.
So is this your bench, Charu?
Yes, this is my bench. As you can see, it's the biggest bench. And another cool thing is we have our own set of pipettes. And nobody can steal it from you because you have name on your pipettes.
Oh, that's nice.
Yes. Let's go and have a look on my cells and what I do generally. Let's go to tissue culture. I take out my 4 dishes where I seeded my cells yesterday. So whenever we set up any experiment, we always need to have a control dishes where we do not give any drug. So these are my control dishes. Let's say 2 dishes where I give them the drug. Now we incubate them for 8 days. We keep checking the cells if they're growing okay, if there is no contamination, like every 2 days. And so it's been 8 days. I had a look on them. They are growing fine, and I think it's time to count them. So we'll just take them out. Let's check if the dishes are dried, and I think they are. So the next step is to stain the colonies. And after you stain them, and you simply then wash them with water. Right. These are the dishes where I added my drug. And since these drugs are very toxic, the cells cannot cope up with it and hence do not grow as well as the cells which were not treated with drug. And now, we can see that there is an effect, right? That the cells are very sensitive towards this drug. But to have an actual number, which is the interesting part, to make a graph, right? So, this is a machine that helps you to count the cells. It is called Gel Count. Super amazing, super fast, easy to work with. So you have your dishes. These are the trays, so you put your dishes here. Yes, so here it says count and we have this number. So you can already see I have my 2 dishes which were not treated, and of course there are more numbers of colonies growing in them, and there are the 2 other dishes where I actually treated with the drug. And then you can see that this is completely full. That means the cells have grown all over the surface of the dish, and here they're growing but not as efficiently as in the other beds.
So I have some few burning questions for you, Charu. What did you show us in the lab?
So it is something that we do regularly in the lab, or at least I do. It is called survival assay. So we give a treatment to the cells and see how well they are growing when you treat them. So in this case, I am giving a drug to my cells and I'm seeing how well they are growing.
Okay. What was the drug that you are using in this experiment?
It is called sumo-inhibitor. So it actually stops a process which is very important for 2 proteins to be able to communicate with each other. And since the proteins are not able to interact and hence they cannot do things efficiently, that's why cells are not growing well if you treat them with this drug. What is the mechanism? I don't know. I'm studying that.
So this is your research project?
Yes.
But I was wondering, we always think that drugs are something useful, good, but here they are killing cells.
It depends in what context you're using these drugs. So some drugs can increase the function of your protein, some drugs can decrease the functions. So this is a very generalized or standardized method. So we stop a function and see how cells are doing. If they're doing fine, that means the protein doesn't have much role to play. But if the cells are not growing well, when you stop the function of a particular protein, that means this protein is important for the cell growth.
Can I make a stupid question?
Sure.
What were these blue dots that you showed us in the lab?
So those were actually colonies.
Colonies? What are colonies?
So cells, if they're dividing over the time, they form these big colonies of cells. It's like just cells making a society.
Okay.
It's like that.
Okay, so it's a lot of cells together.
Yes, so that we can see them when you stain them. Otherwise, if it would be just one cell, it's not visible. That's why.
What are you gonna tell us tonight?
So I'm going to talk about how proteins in a cell communicate with each other and how one protein sends the message to other proteins in a cell.
That sounds cool. Looking forward for that. See you tonight.
So just one thing, if a lot of people are using the internet at the shop, please disconnect because we need the signal for the
So how to move forward? Going next. Okay. नमस्ते, मेरा नाम है चारू और मैं हिंदुस्तान से आई हूँ। मुझे यहाँ पर रहते हुए 2 साल हो गए हैं और मुझे यहाँ पर रहना बहुत अच्छा लगता है। यहाँ के लोग, यहाँ की इमारतें, सबसे ज़्यादा औरतों की इज्जत, समानता और हक। आम तौर पे मैं ऐसे बोलती नहीं हूँ, मेरी हिंदी बहुत ख़राब है और मेरे इंडियन दोस्त ये वीडियो देखकर बहुत हंसने वाले हैं। लेकिन मैं कोशिश कर रही हूँ कि जितना कम मैं हिंदी के शब्द यूज़ करूँ ताकि जो मैं संदेश देना चाह रही हूँ वो अच्छे से दे सकूँ। आप सबको अच्छा लग रहा है यहाँ पर? आपको अच्छा लग रहा है? Anyways, the point I was trying to make is to have a two-way comprehensive communication. It's very important that both the parties understand the same language or speak the same language. That means I can only communicate with all of you if I speak English or all of you speak Hindi, right? Which is not possible, of course. We are in Denmark. I don't know Danish either.
The other button.
It's
The other one. Okay.
It's still not working.
Go. I'll change it.
Okay. Hello everyone, my name is Charu. I'm a Ph.D. student at Center for Protein Research at Novo Nordisk Foundation Centers. Today I'm here to talk how proteins communicate in a cell and why it is important that two proteins or all the proteins in a cell communicate with each other in an efficient way to make sure that cell divides in an efficient way. Before that, let me ask you a question. Do you guys know how many cells are there in an adult human body? Any guesses? Just say 1 or 2 at least.
7 trillion. 50 billion.
You're closer. So it's actually, it's actually 37 trillion cells. Amazing, right? And every day, 1% of total number of cells that you have in your body is being replaced. That means 3.8 million cells per second. Amazing, right? But what amazes me more is we all started from a single cell. How did it happen? Biology happened. It's the only field of science where something multiplies by dividing. I'm talking about cells. So cells divide to multiply and give us what we are today, adult. Think of a cell cycle. It's not working.
I have to. Just look at me.
Okay. think of a cell cycle as this. Maybe not exactly like this. So in science, cell cycle means this. So a cell has to go through several stages to divide. It has to grow big, duplicated its DNA, because at the end it will be divided into 2 cells. So both the cells should have the DNA, right? So before that it has to duplicate its DNA in an efficient way, and then it finally divides. And when the cell is dividing, there are several stages and there are several checkpoints to check if a cell is fit to go to next stage before it actually goes to next stage. So think of these checkpoints as you have security checkpoints at airport when you need to board a flight, right? So the first checkpoint actually checks if a cell is ready to divide, if it is fit. If there is any damage in the DNA, then it is halted. The DNA is repaired, and then it can go again and divide. But if the cell is in very bad shape or it is beyond repair, then it has to die. It is something like this. The good and the bad and the ugly guy. So it depends how a cell is looking, the decisions are made. So the first checkpoint, as I said, is to check if you have self-check-in to board a flight, like a cell is ready. And the second checkpoint actually checks if a cell has grown enough, duplicated its DNA, there is no damage. Just as you are checked in your carry baggage if you're carrying any restricted items. And the third checkpoint is to actually be very sure that the cell is fine, DNA is fine, it has grown enough, and it is absolutely ready to divide. Just as the final gate control you see at the airport, that you got actually the flight number right, you're not at the wrong gate number. And if everything goes right, the cell can divide, and all of you can board the flight. Apart from being checked at all the checkpoints, it is also important that a cell spends only enough time in one of these stages and then exits to next stage, because nobody likes to stand in a long queue to board a flight, right? So these are the 2 proteins which are very important in every stage of the cell cycle. Think of these 2 proteins as these, just to make things easier. And this protein is called cyclin, which is actually holding the cell in one particular stage. And as long as this protein is there, a cell cannot move to next stage. So it has to die, but at right time. So when there is a time that cell has to go to next stage, this protein has to die. And there comes this protein, APCC, which actually have a word with cyclin proteins. And when I'm telling you that they have a very serious talk, and this talk is what a communication between 2 proteins, it should be very critical and precise, and it is more like a death signal. It marks the cyclin protein that you should die now, your job is done. So it actually checks if he got the right protein, marks for death, and the protein dies. And now, since the protein is no more present in the cell stage, a cell can no longer stay in that stage. It has to exit and move to next stage. And then it goes to next stage and so on. But why it is important? Like, these proteins have to communicate efficiently, because if not, what could happen? Either the cell would exit the stage before it is supposed to, and it is not checked properly, so maybe it is carrying bad DNA and it is still dividing, leads to diseases like our favorite ones, cancer. Or a cell will be stuck in one of those stages and there will be no cell division. We will be just like 2 cell or 4 cell, not 37 trillion cells. So in our lab, this is just how 2 proteins communicate efficiently to make the cells division in a good manner. So if there is a good cell, it passes all the checkpoints and it divides. But if there is any bad cells, it is checked, it is halted, the repair is done, and then only it can go to to the division. If not, it has to die, unfortunately. But why is it important to have an efficient communication between these 2 proteins is this. Could be that the good cell which was supposed to divide is not dividing anymore, it is halted, and there are no more new cells. So if you get the wound, it will never be like cured. There will be no more skin, because you need skin cells to grow, right, if you have a wound. So it will take forever to cover up that wound. Or if there is any damaged cell and it is allowed to divide because it somehow like passed all the checkpoints, then it will divide and it will cause the diseases like cancers. So in my lab, we studied this part so that we can understand this part and maybe help cancer patients or patients who have problems with proper cell divisions. And that is it. Thank you for your attention. Namaste.
Thank you, Charu, for explaining us this. And I can already say, like, next time I will be flying or at the airport, I definitely will think about protein division. I don't know if I will remember the details, but I will think about it. I can imagine there are some questions to Charu, maybe. If so, please raise your hand and we'll take them. And if that's not the case, and somebody will, yeah, there's one.
The fact that you don't divide cells and you said like you cannot heal a wound, I mean, then you have a very serious disease and then you die very, very soon, or is it like a common disease? No, not a common disease, but is it like...
It happens, yes.
Yeah, it happens.
Yes.
And then you really have a life expectancy that is very, very short.
Or the life quality is impaired, of course, because if you ever get wound, it will never be repaired, it will never grow skin, and you don't like to...
do something, like if you have a wound and it's not healing, can medicine do something and then you can still live with it, or?
I mean, it can also grow infections, because I heard, like, I know, not personally know, from the internet, I know a case where this person who had this disease, and basically he couldn't go out because we live in the environment where we could have so many infections, right? But if your skin is just open out there, I mean, it is attracting all kinds of infections. So your life is like hell. You're just sitting at home with all the wounds open, and even you don't like to see your open wounds, right? Yeah. So yes, it happens. Yeah.
There's another one. They're all the way back.
The other time we saw some purple colored colors. What colors are these cells? Are they purple?
Sorry? Which?
What color are these cells? Are they purple? The other time we saw some purple colors.
Yes.
Are the cells purple?
Are the cells? No, the cells are not purple, but the dye I used to stain those cells were crystal violet, so it stained the cells.
What color are cells?
What color are cells?
I think they are not colored, they are transparent, cells are not colored. Yes. But we can make them colored, so...
So I think the short answer would be they can be whatever you want them.
Yeah.
I see there's one last question for now there in the corner. Yes?
Obviously, you had to simplify things a bit for this
A lot.
context. And you did a great job. But,
Thank you.
I know this stuff, these mechanisms are super complex. And how are you fishing in the dark entirely, or are there some smoking guns where, like, you know, if we figure this one out, it could be a super big discovery?
I mean, everything is big in this field because every stage, thousands of the proteins which are involved. Of course, I could not put thousands of the proteins here, yes. But there are proteins which are more popular to study and to work on than the other. Yeah, you just need to figure out, and of course, we just study a particular stage, particular process, and then we see which is important and what is important.
Thanks.
In case there would be more questions, we have later like a small talk round where we get all contributors for tonight ready. And if you have any questions at that point, you can post them there. For now, I would check with Eleonora.
Yes.
And ask you, what did you get out of Charu's presentation?
So of course, at the beginning, as you were as she was speaking, I didn't understand anything. So I guess every one of us, unless there is someone speaking the language. So this is us basically. Then she goes and says how many cells we have in a body. That I can't pronounce even this number, so it's fine.
Is it a trillion, Charu?
No, there are three zeros left.
I forget 30.
Yes.
Sorry. It's better. Fine.
No, it's fine.
Sorry. Then we had she told us about the division that the cells has to divide, and then there are different checkpoints that I when we take the airplane. So I drew 2 cells here. That they want to take the airplane that is here, they have a long queue, so I have to go to take that one, but they are old, and so I don't know if they are gonna make it. And also we had here the bad DNA that is inside the cells, and then they should die here. And say, okay, go, you have to die. And also protein, as we understood, they need to talk. So this is trying to be coming back from home and saying,
But we need to talk.
So then you know it's really serious, right?
Yeah, and then we have a question from the womb, but also a question with the cells. So I try to make some violet cells there. Yeah, this is what I got. I don't know if it's right or not, but
No, you got it right. Mostly, yes.
Have you ever seen your work summarized like that?
Sometimes I do, but not as good as she has done. So great job. Thank you.
Good. Thank you, Eleonora, and thank you, Charu, for your talk.
Namaste.
So especially in the beginning, Charu used this idea of language as an analogy on how proteins are working with each other and talking with each other. And I think we should stick with this example for a moment. Because our next presenter is an improv comedian. I'm clearly not, because I just lost my track. And though he is an improv comedian, I know that he prepared a couple of games for us here, or for them to show. Maybe they introduce some of you at least once. And they're going to show us that there's always a way to get a message across. But before we get this presentation or this art performance, I think some of you might already run dry for drinks. So we have a 10-minute break, and then we're going to meet Carlos. First on a small video, and after this we have the performance here on stage. Thank you.
I remember one day, Nobel Prize came out all these articles about it, about gene editing and all the
Have you ever wondered how theater can help us understanding communication? Today I'm here to meet Carlos. He's an improviser at the ICC Theater and also in different theater in Copenhagen. Let's go. Hi Carlos.
Hi.
How are you? How was
good.
today?
Very good, and I'm very happy to see you and to have you here.
Nice, I'm very happy to meet you and to see you too. Is this our stage today?
Yeah, I mean, really everything can be a stage.
So, under this tree, what inspire you this tree?
What is this tree inspiring me? So anything can be an inspiration, and that's the beauty really of improv, that you can be inspired by any single thing. That's why you ask the audience to give you a word of inspiration, because you can work with anything. You can work with a tree. The stage can be a tree. The stage can be but also the theme of the performance can be anything at all.
Hi Carlos.
Hi.
We finally found a place where to sit. How is the communication in a theater?
Yeah, so in an improvised theater, there's two levels of communication. There is the communication between the characters in the play, right? And then the communication between the actors. And this is really what hooks the audience in, because you can see that the characters are saying something to each other, but there is all these high stakes of the actors not knowing what's going to happen next. You can see the character tell the other character that they want to go on a coffee together, and that's something that you might hear normally every day anywhere, and doesn't have stakes, doesn't have, like, there's no tension there, but you can see that maybe because of what the situation that the actors have created, there is tension, also not knowing where they're going. There's so much tension and seeing where is the scene gonna go, what are they gonna do. They don't really know yet what's gonna happen, and yet they're there in the stage.
So not knowing what's going to happen is one of the things that you enjoy more in doing improvisation?
Yes, very much. I think it helps me a lot being in the present. Feeling okay with uncertainty is something very difficult, and especially when you're in front of a lot of people, right? I mean, when you're in front of 100 people in the audience, not knowing what to do, not knowing what's gonna happen, and still being okay with it is something very difficult, but also very rewarding.
How do you work with miscommunication in theater?
Great. So theater is a bit different. It's very different than science in many ways. Whereas when you have in scientific research, I've been a researcher myself. I've done science research, and then you have to know that when you're dealing with science research, you have to be very specific. You have to be very committed to detail, and bring in everything you bring. All the facts have to be based on data or citations, right? So there cannot be any miscommunication when you're doing scientific research. Whereas in theater, it's part of it in a way, especially in improvised theater, because there's gonna be miscommunication between the characters, and that brings a lot of fun and joy and laughter to the audience. And even if there is miscommunication between the actors, that can happen, you can still use it. And you can still, if you, at the end of the day, kind of get together into the same boat, and you manage to overcome this miscommunication or this challenge that you face together, it's really rewarding for the audience and for yourself to have managed to solve this miscommunication.
How are you going to bring all of this improvisation tonight to our audience?
So tonight I'm going to be on the stage with a dear friend of mine, a wonderful improviser, and together we're going to make some exercises to bring to the people in the audience like ideas about communication. So what happens when we have challenges in communication? What happens when there is misunderstandings? And what happens, and how can we move past these challenges and really still connect with each other and still understand each other? And also, what can we do to summarize very big amounts of information in very little time or space?
Sounds amazing, Carlos. I'm really looking forward for that. See you tonight.
Bye.
Thank you. So yeah, thank you all for being here today. Yeah, that's you're coming out to hear science and art or theater, and
I was coming.
I think if the projector is not cooperating with us.
No.
I think it's a problem of communication in the Wi-Fi.
Can you just press to the next one?
It's just more improvisation.
I mean, I can talk without the slides. And I will actually do it. I'll talk without slides. Until they come. So, great. They just needed to be threatened a little bit. Great. So I'm going to talk to you about communication. And you might be thinking to yourselves, what do I, Carlos, have to tell you about communication, right? In an event where we're surrounded by scientists, what do I have to tell you about it? And I really have a diverse background. I've been a radio host, I've been a science researcher, I'm also a storyteller, I'm a published author, and I also am a theater performer and teacher. And I'm gonna work today, I'm gonna try to talk to you about communication in the environment of theater. These are photos of improvised theater performance. And it's mostly what I do, improvised theater. And improvised theater is a kind of theater where you go into the stage with no script, you go with nothing really prepared, and you still create a show. So everything that happens in the stage, on the stage, and everything that happens is something that exists only once in time. It only exists that specific moment where it happens, and it will never be repeated because there's no script. It happens there, you create it with the co-performers and the audience together, and it will never be repeated. And that's really the beauty of it, and that's really the beautiful thing that we might do today here is to create something together. And what I think something I really like, and you saw in the video about improvised theater, is that you don't have script, right? And in a way, we don't have script in our lives either, right? We live unscripted lives, and we still get go on with it, and we still manage, right? When you talk to your family and when you talk with your friends, you don't have the script beforehand. It will be very awkward to people. And you still manage, right? If you like somebody maybe in the bar, you like somebody sitting across and you want to talk to them, you don't sit down and write down what I'm going to tell, right? And it's still, even if you did, for whatever reason, things will happen that will treat it different ways and you will have to adapt and work on, right? So in communication, there's 3 main lessons that I want to share with you from what I've been learning in my years of international theater experience, and that I think can very much be extrapolated to our day-to-day communication, something that we can learn that will help us anyway in your unscripted lives. Great, so there's something about these challenges that we face. We're gonna have challenges, we're gonna have miscommunications, and we can still go through that, right? We saw in the first presentation in biology, if the cell doesn't send the right message, it will not work out. But in life, if you say something that is off or if you miscommunicate with each other, you still might be fine, right? We will see what happens. There's also difference. There's different languages. You might face somebody who has a different background than you or speaks a different language, and you can maybe still communicate. We will see what happens. There's also something about the context. We all work within a context. When we're communicated, we have our personal context, our cultural context, and we have to be aware of other people's personal and cultural context as well. Right? So, but I'm talking here, and I really kind of just want to show it to you, right? Because why not? Since we are here and there's a stage, why not just go for it? So have with me the next slide. Have with me Erica, which is a wonderful performer, and was gonna come up with me into the stage. So we have a piece of a few scenes, a few kind of improv scenes and kind of games for you. And yeah, to get started in the first one, we just need 2 volunteers to help us. We don't have to do much. Just sit on the side of the stage. So please, yeah, somebody from the background. You don't have to do much. Just it's a scientific. You will be helping scientists. So, like maybe. Yeah, or maybe somebody.
We're gonna take good care of you.
We will take good care of you.
There's back here. Come.
Yeah, please. Yeah, come in. We need 2 people. So
I was like, hey, you got one. No, we need 2.
Great. So we have 2 people now, and we are gonna create a scene here in front of you, Erica and I, where every now and then we will kind of forget what we have to say. So we will just talk in the shoulder to our dear helpers and audience members, and they will finish our sentence or say 1 word if we're speaking for hours. For example, I can say, really the most important thing that happened to me today was
to get here and talk to all of these nice people.
Great. Nice. But it will be we will be in the context of the scene, and I just trust you. You can say whatever you want. Just don't be offensive or inappropriate. I trust you're smart, sensitive people that will still, yeah. So great. And to get started, we need a suggestion from you, a word of suggestion of a place, maybe a location that could fit here, like an ice cream shop or
Botanical garden.
Botanical garden. Thank you very much. Hey.
Hey, hey.
You know, I really enjoy the fuzzy leaves.
The fuzzy leaves?
Yeah, they're very tickly. Yeah.
Tell me more about it. Tell me what's your favorite?
Just touch it.
I don't know if I can do it.
It's completely harmless, trust me.
This is a botanical garden. There's can be anything here, really. I mean, what about the poisonous, fuzzy leaves?
That's the beauty of it. There can be anything at all. You could inhale something, and next thing you know, you're seeing potatoes.
Are there potatoes in this
botanical garden? Like, why are potatoes underrated? They don't ever get into botanical gardens.
Marisa, I was actually hoping today we will see some potatoes. I mean,
You were?
I thought I would take Marisa on a date and we'll go to see potatoes to the botanical garden.
Oh, this is a date.
Well, we can also call it
a meeting of occurrence.
We can just call it a meeting of occurrence. You know, like two people that don't have any specific interest on each other.
But they both like potatoes.
They haven't brought any flowers to each other.
They haven't. I have.
I have too.
Oh no. The flowers I bought you are potato flowers. Potato flowers!
Oh my god. That's just like my favorite thing on the world are potato flowers and
you.
And you.
I've never looked at myself through a microscope or, a magnifying glass, but I looked at a potato flower through a magnifying glass and...
Can I see?
Yeah. You can come closer.
This really just remind me of...
Something very awkward.
Something very awkward.
Doesn't have to be.
It is.
Well, when I feel awkward, what I like to do is ride ponies. Ride ponies.
That is such a great coincidence.
Yeah.
Because this botanical garden / pony pony company.
Oh my god.
Yes, but I don't want you to think that I don't know you. Like I've been just preparing this like casual botanical garden.
So casual.
And what I really just need to tell you is
your examination tomorrow is cancelled.
Tomorrow is cancelled. I mean, the world is ours. We can go out and do whatever we want.
Oh my goodness.
Yeah.
Did you?
I cancelled it myself. I sabotaged the examination.
I was wondering.
Yeah, I went there and just...
Ride ponies.
I went there, started riding ponies, and they were like, this is crazy, we have to cancel the examination. I mean, what?
Oh my goodness, well there's only one way to celebrate. Let's ride some ponies. Come on, take your pony.
Whoa. I didn't want to say it, but it's the first time I'm riding a pony.
Doing it with one hand.
I just thought it would look cooler.
Cooler.
Do you know how to brake?
We don't have to.
We don't have to.
No.
We can just continue riding into the distance. And
Both literally and not so literally.
I love the metaphors. You know what? If I had to summarize this day in one word, I would say
turtle.
Turtle.
I really came out of my shell today.
We're gonna clap Erika out of the room. This is because we're gonna play a little bit of a guessing game, and in this kind of game, this scene, she's gonna have to guess 3 songs. So I need from you, dear audience, to tell me songs that are well known that both her and I will know and that you would like to hear about. So please just one song like Hakuna Matata or, yeah, please.
Your song, Elton John.
Your song, Elton John. Okay, great, thank you. Another one, please keep it coming.
My heart will go on.
My heart will go on. Okay.
Mamma Mia.
Mamma Mia, perfect. Great, yeah, we got that, right? Let's clap Erica back in. And
All right. This one goes out to my favorite pony, Cinnamon.
Well, I have to say that for what I'm gonna do. I'm gonna
Sorry.
mime to her the songs and she will guess the light singer.
I'm singing already. Tonight we are I am and you are you, my pony Cinnamon. You are you, we I am, we are you, your legs are the prettiest legs I've ever seen. They're tiny but they're strong, and I like to sing you songs. And I have written this song for you. I call it Your Song.
When Erica getting closer, please do clap or like.
Yes, please.
You know, getting closer to find out. So I've said that before. Thank you for that. See you. Great.
I have another song and another pony to sing it to. Sweetie Pie is her name. Okay. My heart is the size of my chest. Sometimes it's difficult to breathe, but I still have a heart. So it beats. It beats far. It beats running. It goes really fast when I'm running, and my heart is running. My heart is running away. My heart is walking. No, it's not. La la la, march to the beat of your heart, my little pony. Walk, walk, walk. March, march, march. Go from one place to the other place. My heart will go on. Do I have a third pony? I do. And it's called Butterscotch. I've had you since you were a baby, a little baby girl. But I cradled you, I hugged you, and me. I raised you to the sun, like the Lion King you are. Because little babies need to grow up and get born. Sometimes they're born to be wild, but not you. You, I, baby, the two of us, me and you. You are a baby, you're a baby. I'm not a baby, I'm a baby. No, 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 no. I have very long hair and I'm fabulous, and you have longer hair and you're fabulous. I'm a princess and I give birth. Oh, it's me, I give birth to myself. Nothing ever matters anymore. Because Italians are taking over the world. explaining things. Saying things are excellent. Excellent. But why? Why is this so excellent in Italian? We're making some gardening. Or maybe we're making some wine. No, no, my little pony, this got very far from us, but we're Super Mario Brothers now. Mario, marry me? Why, Mario? Why? Nothing matters anymore again. Okay, let's take a breath, my pony, and let her hair down. Hair, baby, mother! Oh, I am your mother, my baby pony. But I'm your tiny mother, I'm your mom. Mom. But why, Mom? Mom, why? Mom, why? Mom, tell me why. Mom, 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 mother, mom, mama. I don't want to die. Mama, 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 I am your child. Mama, 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 mama. Me, mama, me, mama, me.
You can see, even when there's challenges, you can still make it happen. Great, so we're gonna do a last kind of scenic thing, and we need from you, we need that somebody tells us about a movie that you've watched, that you've enjoyed, or you haven't, and just scream out movies that you've watched, or you like. Something you would like to see on stage.
What we do in the shadows?
What we do on the side in the shadows. Great. Oh, yes. Great movie. Taika Waititi is the director, right? So can you tell me something? I need you to tell me what is this movie about, because documentary about vampires. Documentary about vampires. So what happens? Kind of a little bit chronological. Who are the main characters? Can you tell me that? How many vampires are main characters?
And what's happening there?
If somebody else has watched it, you can also chip in.
4 vampires and their roommates.
4 vampires that are roommates.
They're New Zealanders.
And they're New Zealanders. Great. What about what happens? What is really the main thing that happens in this movie?
It's a show about vampires living in present day, but they're inspired from different eras of movies. There's like Nosferatu vampire.
Nosferatu. What is the other one? Tell me another era of time that can be another one.
60s, 90s pirate, vampire.
Vampire from the 60s, 90s. Okay. The third one. I think we've got.
One of them is dating a human.
One of them is dating a human. It's
an old lady.
An old lady. One of them is dating an old lady. Great. And what about there's any quote that you can remember or you can think that they maybe said in the movie that you can.
I'm a very bad person to start something because I have very bad memory.
We'll use that.
At some point they get in a gunfight with werewolves, and some of them get quite vulgar, and they say, "Oh, come on, we're werewolves, not swearwolves."
Not werewolves, not swerwolves. And that one, are we werewolves, not swerwolves? And I'm a very bad person to stand something because I guess
That's fine, we'll take it.
More than that, I cannot do anything about it. It's in the script. So what, can I know what happens at the end of the movie? Is there some closure that we can kind of play with that? Well, I mean, yeah, it's a spoiler that we can accept. Is there something that happens at the end that you're like, okay, now the movie starts? Maybe some of you have watched this over here?
I would want to say that they go back into their coffin, but I don't know.
They go back into their coffin? All right,
Okay,
we'll take
great.
that. So we're gonna, I think, honestly, I mean, movies in Hollywood and also in New Zealand productions, I mean, they take 2 hours to tell a story, and we live busy lives. We don't have that time. That kind of time only nowadays, really. So we are gonna do you a favor for the ones that haven't watched the movie. We're gonna reproduce that movie in 1 minute. And I think I have a stopwatch here, and I think we can all trust Sebastian there. You can maybe keep time for us.
I can try.
Yeah, and you can keep like just 1 minute. We're gonna do this movie in 1 minute, and you have to say time when it's...
I can do that.
Great. So, thank you very much. And while you prepare it, so I'm just gonna go a little bit over it again. So there's 4 vampires that live in a third flat apartment, you said, in New Zealand, and they have. There's one from the 1690.
He's just very sexy.
He's very sexy. There's one very sexy. There's a very sexy vampire, and there's one that is Nosferatu-like. And then they find some werewolves, and they say, "You're werewolves, not werewolves." Then one person at some point says, "I am a very bad person to start something because I get into Paris." And yeah, I think
Then we go back to
one is dating a grandma, and then they get back into the
Yes.
Okay, we got this. Great. When you're ready, just say start, and we'll start.
And go.
You should really do the dishes more often, Mitch.
To go and see my grandma girlfriend.
You always get out of responsibility.
Fuck the dishes. Hey, darling. Oh no! Oh shit, there's some werewolves over there. Fuck you! Hey, wait, I thought you were werewolves. No, it's werewolves. I think we have to go back into our coffins.
But I'm not ready. I'm still looking myself in the mirror.
Time! I mean, I honestly, the movie was there, clearly, we all saw it. Honestly, I mean, we can do this much shorter. We live even busier lives than 10 seconds ago. I mean, we can just make it in 30 seconds, right?
Yes, definitely.
Yeah, let's do it in 30 seconds. Yeah, 30 seconds. Yep, you say start. Yeah.
You should do the dishes more often.
I have to go and see my grandma girlfriend. Darling, no, why did I do that? Holy shit, there's such werewolves. Fuck you! Oh, you were werewolf, not as werewolf. We have to go back into our...
But the mirror. I can't even see myself in it.
Yeah, you know, I mean... Too long. I mean, I feel like we can just sort it out and then it's still there, right? I mean, we can... you think we can go 15 seconds, right? I mean, yeah, let's... Yeah, I mean... Yeah, let's go 15 seconds. Okay, start. I have to go my grandma girlfriend. Holy shit, there are some werewolves. Fuck you, werewolves. Go back into the
mirror.
Time. The problem is that we even have like spare seconds at the end, so I mean it's only fair to this audience to go. I mean I don't know if it's even possible, but we should go into 7 seconds, man. Right? Yeah, you say start, we will do it. Start. I see my grandma girlfriend. Fuck you! Time to bed. Okay. Can we even do it, Sorte?
Yes.
Is it possible?
How long you want?
Is that even measuring 3 seconds? I think it can do. Well, pushing science right now.
We are right on the limit.
Okay, we'll do it. 3 seconds.
And go!
Time! Bed! You defy the limits of time. And, yeah, I mean, we're still going to... Yeah, I mean, do you want to see 1 second movie?
Yes!
Okay.
Do I need to time that?
Yeah, 1 second.
Are you ready? And go!
Grandma, go! I haven't finished.
I'm sorry. I'm so sorry.
I want to bring some conclusions from what we thought, right? For what, yeah, I mean, this was very enjoyable, I hope.
Yeah.
I had a great time performing here with you. You're a wonderful audience, and thank you for all the participations. It really made the show be. Otherwise, we wouldn't have anything to do. And I want to kind of bring some ideas from what we see. So when we face challenges, I mean, when we face miscommunications, and we're going to face them, I mean, let's be real, there's going to be miscommunications in our lives and challenges. And I mean, we should still want to adapt, and we should still want to overcome it, and we should still just acknowledge them. We say in improv that mistakes are gifts, and that's also why I tried to take this extra suggestion. Mistakes are gifts, and we should take them and use them, and I don't know, like, it can draw a scene forward. It can also be a very nice inside joke that you can have in your life when you make a mistake with somebody. You to enjoy that after. It brings you closer to the other person, and also it really is something to overcome, and you learn from yourself and you learn from the situation. So that's what happens with challenges, and also differences. When we have differences, we were here. One person was singing, and I was like miming my ass off on stage. And really, I mean, we don't talk fully the same language, although we still can communicate in some way, right? And when you're gonna be out there, you're gonna meet people from different backgrounds with different ideas, different ways of communicating, different ways of understanding, and you have to be mindful of it, about it, and you should also be open-minded about it. You should be mindful that there's gonna be people with different ways of communicating, and just embrace it and try to take it as a learning opportunity, right? And then about context, in the last game, you can really see that if you share a context, if you share a set of like knowledge with other people, you can really summarize a lot. So because you had seen the previous 1 minute 30 seconds and so on, you saw that 1 second movie and you were like, yeah, it's right there. So when we have to be mindful of the context that we have, the knowledge that we have, the context that we have, personal and cultural, but also we should be mindful of the cultural context of the people we met and encountered. So if they have the same context as you, it's easier to communicate and you should use it to your advantage. But if they don't, just be aware of it. Yeah, we can go to the next. So I just want to say that communication in general happens. It happens if you're willing to communicate. If 2 people want to communicate, you will end up communicating. There's many ways to do it. If you're vulnerable and you're accepting and you're open-minded and you're not judgmental, if you're not judging, if Eric instead of continue singing will just judge my mind, we will have work, and she just continue and she got it, right? So we should be open to adapt, trust each other when we talk, and we should seek understanding and be willing to understand each other. So yeah, that's my presentation. Thank you very much.
Well, that was quite an illustration of where communication works great and maybe not so great. I will long remember Mamma Mia. I think you will too, Erica. Mamma Mia, Stigma, yeah. Eleanor, I think there was a lot of stuff for you to pick up there.
Yeah.
So I'm actually quite curious to hear what you came up with first.
Yes. Are there questions for Carlos first?
Yeah, is there any question or something you want to ask, or something that, yeah, you will have any further questions about our performance of...
You're in the back. Yes.
Well, not about performance, but it seems like you communicate all the time. How do you deal with that little voice in your head that tells you that maybe people I'm talking to don't understand what I'm saying.
Yeah, that's a great question. Like, so when you communicate, when you're talking with people, and this voice in your head that sometimes you self-judge yourself, and you're like maybe they don't want to hear what I'm saying, or maybe this is not really. I should just maybe not talk. And I think that's part of really what you work with when you do improvised theater, is working with your judgment and try to like set that basically. Because if you're having to talk and act fast and react in instincts, basically, you have to really trust it, and you have to be mindful that most of the time your brain is going to take the right decision, and if it doesn't, you're still fine. Like, it's okay, you will get there anyway. So I think you should take it as an opportunity to get closer to the people you're talking to, and also to learn about yourselves and from them. Hope that helped. Cool.
If we want, we have time for another question. But also here again, we also have the time for this later. But are you spotting something? There's one now.
Yeah. How did you get to do like improvisation? Because you had different backgrounds, and so how did you choose that?
So when I had done performance and like a theater throughout my life, and in Spain I had done it a lot. And then when I came to Denmark, for whatever reasons, I ended up being a radio host in a radio show in a little radio in the north of Copenhagen. And I was just having this weekly show for music and books and movies and so on. And I really enjoyed talking with the hosts and with the guests and just exploring different environments. And one day there was this guy that had a radio show about improv theater, and I was so curious about it. I talked to him and I was instantly hooked. And I started doing practice, like just signing up for courses and doing practice. And I traveled all over Europe and also to the US, to Boston or Chicago to learn more and to just grow into the performer actually. Yeah.
Yes. Thank you for this answer.
That's a good question. Yeah. Yes.
Then I think I can start.
Wow.
And
did you have enough space?
No, I didn't. But I tried to squeeze all the stuff. So this is Carlos on a stage, and also at some point he mentioned that we are living like live, and we don't have script. So this is Carlos' family with a script in the background there, and then we have the first 2 years, the two like people that like came here and help you to make the first game, and I think Erica started with sniffing a flower or something, and then something was poisoned there, and there was a potato. And at some point they were talking and going around with a potato flower and a magnifying lens. At some point it was getting so strange, and apparently a pony appears that say that there are no exam tomorrow. So I guess, but at the end, there was also a turtle. I really don't know. I put a what because I have no idea why a turtle was there. But anyway, this is what I got from that tool. Then we have the second game where Erica was singing. I'm sorry, you're like, I had few time to draw you, but like my heart will go on, your song, Mamma Mia, and then you sang it to your horse, your pony. And at some point there was also a child. There was a princess, something there, so like, and then there was the why for the Mamma Mia. We have to get it at some point, you know? And then the third game, which, I mean, I don't know the movie. I have no idea about this movie, but there was a...
You've seen it now, right? It doesn't get better than this.
True, I saw it. Then there was like a vampire. Then we have Busy Life. And then I got there was a vampire with a grandma here. Then coffins. Then I get a werewolf or something, like somewhere. And that we were making the 30-second, 1 minute, 50-second, and then I didn't have much space. And then I really like also the end of the talk where like mistakes are gift, and then we have all the cultural background and all this stuff. So I think I got a bit, a lot of stuff to draw there. And that's all.
Great. Thank you very much.
Hard
like to follow upon? No, I really liked what you were saying in the end, that the movies worked so well because we have a common reference point. At least we all know what a movie are, we all know the song somehow, and at least some people in here also knew the movie that was playing out here. But at least the question, what's happening if we don't have this common frame? And in science, that's actually the case. Science is on the edge, by definition. Science is there where there's no more knowledge. So scientists have the task to figure out something where there is no contextual information. So I'm training, I'm a geologist, and one of my geologist professors once said a beautiful sentence, well, geologists are really good at making a lot out of very little. And our next presenter should be really good at this, because he's not only a geologist. In one talk I had with him, he called himself actually a space geologist. And that's really a title that I really envy. Job description. And his work is to make sense out of things that are very long ago, we talk about millions of years, or things that are very far away, we talk about light years. And he will explain to us how he's doing that. That's a very vast topic, and before we make a lot of sense out of very little there, I think some of you make a lot of drinks out of very little drinks. Thank you for this one. So we have a 10-minute break, and then we start again with the video about Georgi, and after this, his presentation here.
Have you ever wondered what kind of message can we get from a meteorites? Today I'm here at the Natural History Museum of Denmark to meet with Georgi. He's a Ph.D. student at the Globe Institute, where they do a lot of cool stuff with meteorites and analyzing rocks coming from the space. Let's figure it out. Hi Georgi, how are you?
I'm alright. How are you?
I'm fine. Coming to meet you. What is this?
Some meteorite from Greenland.
What kind of meteorites it is?
That's the meteorite where every Friday at 4 p.m. we meet downstairs at to go for beers.
Okay, this is the importance of this meteorite.
Yeah, it's a landmark here.
Are you studying this meteorite?
No, I don't study this one, but I study a lot of different iron meteorites, yes.
Can you bring us there?
Yeah, sure.
Let's go.
Perfect. Hello, so this is where we start studying every single meteorite, because before we can do anything with it in the actual lab, this is like a sample prep lab, like it's a pre-lab. So before we can do anything with it in the actual lab, we need to get a small piece of it. So what I do here is I study, for example, iron meteorites, like the one downstairs, but I study different ones. And what I do with them is that I take a meteorite, and then I need to get like a small piece of it, like this one. Small piece like this, small, tiny, shiny, cute iron meteorite.
That's super cool.
Yes, and so I need to get a small piece like this, like that. And for that, we use something like this one. This is a wire saw that has a wire and it can saw. So after we get the small piece, we go to the clean lab, and let's go there. Clean lab number 3, yes. We have, I guess, 2 more. Yeah, so what happens to the meteorites here? So I showed you some small pieces of meteorites, and that's the next step that happens to them. We basically need to make them into liquid. So every single beaker here is a meteorite.
This is meteorite soup.
I guess.
But how from a solid sample you can get this liquid?
Yeah, so we use acids. For this we use acids. So this whole thing happens here. So what we do here in our lab is we do isotope analysis, which means we need to. So we basically use some small. We use some traces in the meteorite it's basically like cosmochemical DNA or something like this. We use it to fingerprint planet formation, to understand which planets relate to which building blocks of planets. We need to separate one particular element, one chemical element from a meteorite, the one we want to study. And we do this using those things, which are called column chemistry. And for example, I study nickel. And so yeah, here you can see the recipe for this one. So this is the recipe for this chemistry. So basically what happens is we put a small drop of meteorite here, and then this drop of meteorite goes through this. It's like organic compound that reacts with different chemical elements. And what happens here, that nickel goes through, but zinc gets stuck. So basically, yeah, I put my sample in there. By the time the samples go through, only nickel goes through. Everything else in the sample gets stuck in there, like stuff that I don't need, iron, zinc, or whatever. And then we end up with something like this. This is purified nickel from a meteorite. Once we have this, we can measure this nickel, its isotope composition on mass spectrometer.
We saw some sample in the labs. What they can tell us about Earth?
Not much really. I mean, every single meteorite is not enough to understand anything really. But once we get a lot of them, once we have all of this data, like once we have a lot of different information about different meteorites, then we can compare planets and try to understand from what kind of material in space they, like that there is in space, from what kind of material they formed.
So, Georgi, what are you telling us tonight?
So I'm going to be talking about, I'm actually going to be mostly talking about what my colleagues here are doing, because in the end, I'm gonna talk a little bit about my stuff, about the meteorites and using them as to fingerprint play information. But what I'm gonna start with is talk about all my colleagues in this Center for Study Play Information and what they are doing, what their different ways to study play information.
Okay, that sounds amazing. See you tonight.
No.
Yes.
Yay. Hello. Does it work? No. Yes, this one first. Hello, everybody. I'm Georgi. Yeah, I guess you figured out who I am. I'm Georgi, PhD student at Center for Star and Plane Information at the Copenhagen University. And so, to start with, this is Earth, if you didn't know. This is the moon. This is like one of the, I don't know, iconic pictures when astronauts first flew to the moon and they're like, wow, Earth is so far away, so small, let's take a picture. And now it's recognizable. Before we start, I'm going to ask you a question. Does anybody know here how old the Earth is? Any ideas? Not from cosmochemists. What? 4.5 billion. Yes, that's roughly true. But do you know, how do we know this information? Isotopes, yeah, kind of. But the thing is, we don't actually know the age of the Earth. We know the age of the Sun, and all we know about the age of the Earth is that it's roughly as old as the Sun. And even worse than that, so we barely understand when the Earth formed, we even worse understand how it happened. And so if we want to study, if we want to figure out how planets form, what's the most obvious way to do this? What's the most obvious and simple way to try and figure out how something happens? Yeah. I mean, or we can just look. I mean, I don't know, if somebody wants to understand how cocktails are made, I can just go there and look at the button and like, look at it. So yeah, we can look at stuff. And this is roughly where, I mean, this is a big, we call it giant molecular cloud because they're giant and have molecules. And so this is where star and planet formation happens. And I mean, so stars and planets form together. It's all one process, really. So it happens in all those huge clouds. And as you can see, like this is one star, one star, one star, one star, one star, one star. So it's really big thing. So we actually need to zoom in. And the way to do this is using this thing. It's called ALMA large millimeter/submillimeter array. It's an not Alma. Alma is the whole abbreviation. Atacama, it's in Chile. So the thing is, if we want to look at the, so planets form, like one of the most important components to form a planet is some kind of solids, dust, space dust. And when we look at this, actually, like if I don't know, if we have a fog somewhere or dust storm or something, we don't really see through much of it. We just look, okay, we see some, like, okay, yeah, there is a fog there, and we see like the first several, like the front of it, like the first meter of it, but we don't see through it. So if we want to see through it, we need something with a longer wavelength than visible light. And for that, we use, so in this case, it's far infrared and microwaves a little bit. And fortunately, because if we really want to have high precision, not precision, resolution, if we really want to zoom in on something far away in a good detail, we need a really, really, really big ass telescope, whatever. And fortunately, unlike with visible normal telescopes where we have lenses, cameras, and stuff, with far infrared and microwaves, it's a bit easier because we can actually have a lot of different dishes, put them all of them together, and pretend like it's one big telescope. I mean, we obviously get a slightly worse picture than if we had one huge dish, like a radar basically thing. But yeah, I mean, this is way easier to build, and all we need to do for that is to have some more computer power going on to try and combine the picture into one thing. I have no idea how it works, but yeah, it's complicated. And they have somewhere, I don't know where it is, but like you see those big roads. So they have like one big truck that moves them around to create different arrangements, so to pretend like it's a different big telescope every time. Okay, so once we do this, once we zoom in on one young protostar with planets forming around it, we get this picture. So this is again not visible light. This is in far infrared, which means the brighter, the more dust. Yeah, that's how infrared works, I guess. And so here in the center we have a star. We don't see it really, but it's in the center. And around it we have a disk of dust. Oh, there is also gas, but we don't see it in these wavelengths. So here we just see dust. And this whole thing is called protoplanetary disk. So it's disk where planets form, I guess. We don't know really if they form there. And you see those gaps in the dust. So those gaps in the dust, this is what we think are gaps caused by young planets that are forming and just carving a gap in the dust around it. But so if you look at this picture, if I look at this picture, there is not really much detail going on. Like this whole thing is the size of a solar system. So it's huge. Like if Sun is here, the Earth would have been somewhere here, and then maybe Jupiter somewhere here, and then like Pluto somewhere there. Like this thing is huge, and I don't know, like whatever, 1,000 pixels or something. Not a lot of detail. And so this is a map of a galaxy. This is very different from protoplanetary disk. So we just looked at a picture of a disk around a star where planets form, and now we look at the whole galaxy where we live. This is not a real picture. This is a map. It's drawn by an artist together with an astronomer. And we have Sun here. We have Sun here, and with those microwave infrared telescopes, we can see roughly this area, like really only this. I think it's roughly what we talk about there is what we can see is like roughly 1,000-ish light years. But the whole galaxy is big. The whole galaxy is huge. And most of the star and planet formation happens actually in the center where there is more stuff going on. We literally live on the outskirts of a galaxy. So what we just do, so basically what people do in there is imagine, again, trying to figure out how cocktails are made. But all you get is an iPhone, and you need to lie down, look down there, and like you can take one picture, probably through some blanket or something, because it's actually, there's a lot of stuff between us. And from that, you need to figure out how cocktails are made. It's hard. I'm pretty sure. Okay. What other way to figure out planet information? And somebody before that said time travel. So almost. We have a planet. We species living on a planet can literally, to figure out plane information, can actually maybe like just try look at our planet and figure out what happens to that one, how it formed. But the problem with the Earth, I mean, Earth is it's like the most annoying object to study if you're a cosmochemist, because Earth is like we have volcanoes. Volcanoes melt stuff. We have rivers. Rivers oxidize stuff. We have humans. Humans put nukes and I don't know, radioactive waste into rocks. Like we don't get stuff to like it's all altered. Like basically, if we were, I don't know, imagine you are a forensic archaeologist and investigating a crime scene, and you know that a robbery happened, but once you get there, and like you need to get fingerprints, but once you get there, all you get, not all you get, like the whole building just burnt. So that's roughly what happened to the Earth all the time, because Earth is geologically active. And what can help us are those guys. This is a meteorite. This one. This is different meteorites, but yeah, we have meteorites. So we can look at those because those are actually, okay, this is not the same building. This is not the same building where robbery happened, but it may be a neighboring building, and we can, I don't know, try to look into cameras and records there or something like this. Or maybe a person before robbery had a coffee in a coffee shop across the street or something. So that's literally, that's the only evidence in the solar system we get from the times planets were forming. You can pass it around. Just get it back to me somehow, please. It's not a rare, it's not expensive. Like I think it costs like literally 20 kronas, but it has emotional value to me. I mean, it's literally just a rock. So basically, when we're talking about, so we figure out, okay, looking at cocktails made like from here, like looking away from the cocktail somehow. So when we study plane information with meteorites, what we do is we go into bakery, we try to figure out what bread they have on sale, but all we get to look at is crumbs on the floor, and that's it. And worse even that, like, okay, if we send astronauts on the moon and they bring samples, the breadcrumbs we want, specifically the ones we want back to Earth, yes, that's a good analogy. But with meteorites, it's actually, we don't even get to go into the bakery. We just hope that janitor will trash the breadcrumbs representative of the bread in the bakery into the trash, and then we hope that those breadcrumbs will not be mixed with banana something. So yeah, again, it's hard. But there are also many other ways, which I will cover in way less detail. For example, we can send robots on Mars and try to study other planets. Again, with way less detail, because robots are, I mean, you saw the video with my lab, and this is smaller than my lab. It's big robot, but it's smaller than my lab, and it has less capabilities. Or we can do supercomputers. We can put a computer, big computer, this is all one computer. We can put all the physics we know about planets in there, and then run a simulation trying to figure out how planets, how it happens, how they form. But those are not real planets, those are made up planets. We can easily just, oh, maybe we forgot to carry over a minus or something, and then we have, I don't know, planets forming from cats. Or another thing we can do is, oh, yeah, I was told about the shadow there. Another thing we can do is look at other planetary systems, the ones that's already formed. We can use telescopes. And so here in the center, we have a star that's blocked, because if it wasn't blocked, it would have been way too bright for us to see anything. So there is a black screen covering the star. And so that stupid cosmochemists who don't know any actual astronomy know that there is a star, it's drawn there. And then we actually can see some planets around it. But again, not too much detail. Yeah, not much. Like that's a planet. Yeah, and you see how long it takes to make a GIF like this. Okay. So this is the world of plane information studies. And now I'm gonna talk in more detail about my small, tiny own world of specifically cosmochemistry plane information studies. specifically what I do in the lab and how it works actually. Because I mean, in the video, what you mostly saw, it was like pretty pictures of me in a lab coat that's like made of cellophane or something, but not actual science. So how it happens? Whew, I need to take a breath. Where is my beer? Thanks. Oof. So this is a diagram that's roughly showing what overall is happening. So we start with a cloud. We start with a big, giant molecular cloud. Then somehow, in some part of it, it's dense enough that there is a star appearing. And first, the star has like dense part of the cloud around it. And then finally, this dense part forms a disk, the one we saw in the glowing orange picture. And then somehow, after this stage where we have a protostar with a disk around, we end up with a star with planets around. And two main ideas that roughly cosmic chemists and astrophysicists and people in general have about plane formation are, so it either happens roughly on this stage, early on, during the disk stage, from the dust that there is around, or it happens later. So dust first forms asteroids. I mean, okay, we don't call dust dust, we call dust pebbles for some reason, and then we call asteroids planetesimals for some reason, but it's really the same thing. So either here from dust, or later on, dust first forms asteroids, and then asteroids collide with each other and form planets. And yeah, picture. Star, forming growing planet, a lot of dust around, and this happens early on and fast from the dust. Or star, growing planet, a lot of asteroids colliding, happens slower from big asteroids. Huh. Fortunately, when we look at the meteorites, there are, if we make the most simple, primitive classification of meteorites, and we try to classify them in two different classes, it's going to be two. Wow. If we try to do two things, it's going to be two things. We're going to get primitive meteorites. Those are literally just piles of space dust. So nothing happened to them. Like once the dust formed, got into one big rock, nothing after that happened. So we have here like those small round things that are called chondrules because they're small and round, and from Greek it means a grain. Then we have those white things that we call refractory inclusions, and then we have like those black stuff, which is literally just space, dirt/dust. And then we have, so those meteorites are really representative of the dust in the disk. And then we have those meteorites, the ones you saw mostly in the video because I'm studying those a lot. Those meteorites are really representative of asteroids because those are called differentiated. And so it means that they were in an asteroid or small planet that was hot enough at some point to melt, so that we have a metal core, like this piece. This is probably a piece of a metal core. Actually, no, but it was melted somehow. It's just not particularly cold, not particularly this one. And then we have also a stony, rocky mantle around it. So those ones are actually representative of asteroids from which planets could have formed. And as you can see, I drew an outline here because it means something, red and blue. So what does it mean? We can take a sample of Earth or of Mars or of, okay, no, we cannot take a sample of Venus because it's really, really, really hard to get on Venus and then get back because of the atmosphere. Okay, so we get Earth and Mars basically, and that's it. Also Moon, but it's kind of the same as Earth in terms of where it comes from. It's all just. So we take a sample of Earth, of Mars. We take a sample of those meteorites. Then we do all the stuff you saw in the video, and what we get in the end is this blue and red picture of Earth, which means we can try and figure out using those nickel isotopes. I mean, I'm using nickel isotopes. Other people use different stuff. Any elements, I guess, apart from elements with no isotopes. Yeah, so we try figure out, okay, so this planet probably formed from dust and asteroids in these proportions. Or maybe this planet formed in different proportions. And once we get this information, we can actually try and figure out, okay, what was the mechanism that made it this way? Yes, no, too early. So, okay, So this was my small world of cosmic chemistry. So why does it matter in the first place? I mean, you already got the spoilers, unfortunately, but why does it matter? The thing is, like the big question that nobody is really, almost nobody in space sciences really works on, but which is behind any science about space is, are we alone? Do weird brainiac Martian aliens in really tight and weirdly fetishized spacesuits exist? But really, I mean, okay, there are other forms of life. And the thing is, probably, I mean, we're trying to figure out. The thing is, do we, what I was talking about? Yes, so how it connects to planar information. Life as we know it, obviously there is a possibility that there are other forms of life. Like, I don't know, maybe boron polymer-based life, or maybe silicon polymer-based life, or something like this. But the life as we know it, and we are a carbon-based life form living on the water planet, the life as we know it really needs water. Like you can see it's dry, and here it's not dry, and there is green stuff going on. There is life going on there. So, and different building blocks, they actually have different amount of water in them. So for example, asteroids, you saw them, a lot of them are melted. So if we form planets from asteroids, they're probably going to be dry. They're probably not going to be a lot of water. Or maybe we just need to get lucky to get some water if the asteroid was not melted enough or something. But if we form planets from dust, I mean, for astronomer, dust is anything that's solid, which includes water, ice. So in dust, there is actually plenty of ice. And if we form planets from dust, we actually get plenty of water everywhere on all the planets. So once we figure out building blocks, and also we figure out mechanisms, because different planar information mechanisms imply different conditions. So maybe even if we form planets from ice, from a lot of ice, but it gets all evaporated on the way or something if it's very hot. Or maybe it doesn't. We need to figure out that as well. But the point is, if we figure out how planets form, we can figure out how habitable planets form. And if we figure out how habitable planets form, then maybe we get a little bit closer to the understanding if we are alone in the universe. And so yeah, I just talked about like the world of planar information studies, then my own tiny small local world of cosmic chemistry specifically studies, and now I just covered a way bigger world. This is from Men in Black. Way bigger world of just trying to figure out if we are alone here or not. Yeah, thank you.
Don't you want to stay in the spotlight?
Okay, yeah.
But you're free to take a sip of your
Oh
beer.
yeah, thank you.
Thank you, Georgi, for this amazing presentation. I could imagine there are quite some questions for Georgi. And I'm
okay.
In your presentation, you showed an image of roughly the makeup of Earth from the red and the blue. Later on, you said that the blue sample would have more water. Considering how much water there is on Earth, wouldn't the ratio be different?
This ratio was not meant to represent any actual answer. It was just to show the idea behind the studies. I mean, I personally think that yes, I mean, some people call it Copenhagen model, that planets form from dust. So we just all think that in this our research group. But it's really a biased opinion, and it's not really science outreach if I'm not delivering how we do this. So it's yeah. And the thing is, I mean, we think that, I think that, but other scientists don't believe me. So. And I don't believe them, and I have different ideas, and everybody. So that's right, exactly the point everybody is discussing right now in the community, in the scientific science.
Just as a follow-up, the problem lies is nobody has proof.
Yeah, not really. We don't know. We don't know. Like, I mean, I have some kind of proof. Other people have some kind of proof, and all basically we're trying to get. Okay, maybe if we do this element. Or maybe we can start this new project with nickel. Maybe we'll get better data. So we try and yeah, it's hard. It's interesting.
think you gave a beautiful explanation there how the scientific discourse is working, right? Like you have a good idea, somebody else has a good idea, and then you figure out who has the better good idea, right? Are there any other questions? Right? Yes, right there.
I was just wondering, maybe it's a crazy idea, but has anyone tried to detect like DNA or protein meteorites?
The what?
DNA or like protein?
Yeah, there are people studying that as well. And I think so far it got to detecting, how it's called, amino acids in meteorites. There are amino acids in meteorites. There are also amino acids that we see in telescopes in those clouds. But we really don't know if they get on planets in this condition or if they get destroyed on the way. And then on planets we have amino acids and DNAs forming all over again. Yeah. But yeah, there are people studying that as well. It's, as I said, it's way, way, way bigger world of searching for aliens.
Yes, yes, another one.
Maybe a stupid question, but the meteorites that we have on Earth that you can find and study, are they like originate from our solar system, or are they?
Yes. So all of the meteorites that we know come from solar system. There are some small parts of meteorites that some people think, oh, maybe it's actually not from the solar system. But there is really no way to prove that as of now, and it's like I don't know, maybe in 20 years we'll figure out some way to test this. But yes, all the meteorites in general come from solar system, and most of them come from basically inner part of the asteroid belt. So the asteroid belt is between Mars and Jupiter, and most of the meteorites come from the inner part of it. And that's why I meant that we actually don't get to choose. So we study whatever we get. We don't actually get to choose that samples from far beyond. We just hope that the janitor will occasionally throw out some breadcrumbs from the bakery that are slightly better and rarer than the rye bread that everybody. I mean, we already know that there is rye bread in there, right? We're in Denmark. So yeah.
Yeah, because there's so much interest. Yes.
Do we know if the moon formed as part of the Earth, that like part of the same thing that formed the Earth, or an asteroid that came close to the Earth and was?
So somehow weirdly, almost every single scientist agree on a fact on a model where moon formed after a collision of two planets. But there are a lot of details that we can't figure out, and it's So maybe it's two planets collided, but didn't really get destroyed, and then one started orbiting the other, or maybe it's two planets collided, completely mixed and melted, and then I don't know, some leftover stuff that didn't fall back on Earth formed the moon again, or something like this. A lot of details are elusive right now, but we pretty much all agree that it's a collision of two planets.
In case there are more questions, I would put them to the end of our talk, where we have more chance for this one. For now, I have a question to you, Eleonora. To you.
Yes.
Oh my God.
So I guess the first things that you were saying is how old is the Earth, right? So like someone answered 4.5 billion, and someone said that we can discover it with a time machine that I actually drew like a washing machine. Why? I think it was easy to draw. But anyway, also we had a question about how old the moon was formed, like how long and how the moon was formed. And then you talk about like this telescope and things, and then you put all the dishes together, and then you have many streets going around, right? Something like that you say. I remember streets, and then I drove streets. And then you showed us this, which you say that there are some dust there around, and actually the spot and the hole, they are probably tiny planets. So this is a young planet there, staying there nearby. And then you gave like a bit of like discussion of the space. There was also a question on the asteroid here, like how asteroids, like if there is DNA or any proteins there. So that was a question there. And then you talk about like volcanoes also. So this is a volcano here, and then I get a little bit into spacecrafts, like space, the dust from the space, and you mention a banana somewhere. And then I go trapping that banana, and someone is like, cleaning the planet dust there. And then we went to talk about aliens, because I also like aliens, and I tried to make a nice aliens, but it's not that nice. And like also you mentioned like water, that is one of the most important elements. And also we had a question about what's the composition, and that something like what was there and anything. So at the end, we have all your discussion here, I guess.
Thank you.
Thank you, Georgi.
Thank you. Yes. Yes. I could hear there was going quite a lot on at different tables. I don't know if you actually discussed what I posted or if there were random things. Anyways. Before I ask you guys if you have some questions to them, I would like to ask these three the exact same question. What is the key ingredient for successful communication? And we haven't heard from you so long, Charu, so maybe you start on that one.
If you're talking about people, then I think the intention to communicate is enough. But if you're talking about proteins, which I study with, then it has to be very precise communication, very accurate, otherwise things can get really bad and it's not good for anybody. So that's my answer.
Mm-hmm. So precision is important because it's a very small scale, right?
Otherwise it could be misunderstood very easily.
Yeah. Misunderstanding is also something that was a topic in your talk presentation. What's important for good communication on stage?
I think because we have this big difference between the scientific world when it has to be very precise, and in the theater you can be more loose and you can enjoy that sense of like those mistakes. I would say trust.
Uh-huh.
Because in science you gotta trust the data. You have to trust the data. Whereas in theater you have to trust the other performer, and then everything else, like the words and so on, matter less.
Mm-hmm.
So you can go along with mistakes and it's okay. But the trust has to be with the performer.
Yeah. Trust is an interesting thing because you said like you need to have trust in data, and I agree on this one. But you, Georgi, took this trust a little bit from us by saying, well, we don't really know, and there's idea 1 and idea 2. So either communication, like signals from meteorites, or communication between scientists. What's important for this to work and not be just angry?
So with meteorites, it's okay to be angry with meteorites. They don't have feelings, I think. But
have you tested that?
No, I have reasonable common sense. So, but actually, with the trust, I mean, we do have this problem, but it mostly like we don't have enough trust in our data. Nobody trusts our data, and we don't trust anybody else's data. And that's how we progress. That's the main driver to make better data. And then nobody trusts that again. But it's like it's always like this, cyclical in this way. But the main reason why we don't have this trust, I think, is because we get really snippets of we can barely see stuff far away there. Then we can barely resolve differences between different meteorites here. Then we can kind of model something on the computer, and then we don't have coverage. Like we don't have. We need to interpolate a lot. We need to find something between, because our data is disconnected. Like it's like you get every 10th word in a sentence or something like this. It's.
And before we maybe go a little bit deep into this resolution problem, I would like to ask the audience that some interesting questions that came up while you were discussing. Doesn't seem like that. Amazing.
What about boring
questions? There's one. Yeah, boring questions are also welcome, yes.
We were talking about one of the key is like how to observe and when, like know when you observe someone, and like if it's like people communication and like the observation is, and like it's also close to the willing of talking, like you said, and wanting to understand, but like if you know in a specific context, if you know when and where and who you're observing, and how you can, if you're willing to observe and if you're willing to share, and I don't. Maybe we have other topics, but that's like.
No, we find the idea of observing quite interesting, and that's like a question I maybe want to pose to our two scientists here. You are both somewhat of a chemist, like you're a biology biochemist, you're a geologist, cosmochemist, and my question is a little bit: do you see similarities how you capture a signal? So the idea is you need to look into how one protein is transferring to another protein, and you need to look how out of a metal rock, that's what it is efficiently, you get some information out. Can you compare your work somehow? Do you use similar approaches?
I have a question for you then. Do you think your proteins willing to communicate? Because we figured out that willingness is important. So do your proteins willing to communicate? Because my meteorites don't.
That's true.
Yes, so I think it's...
No, with you. Do they will?
Yes.
Do they have will to communicate
Yes,
with you? Okay.
it's a team effort because if you have paid enough attention to my talk, I said when it's the right time that a protein has to die, it should also wave that, you know what, I think my work is done, I need to die. And then only the other protein can come and actually mark it that, okay, you die. So it's the protein itself that gives the signal that, and the signal is a kill me signal, and then only the other protein can come, recognize it, okay, this is showing some red flags, I should go, have a word with it, and then it kills it. So it actually is a team effort. It's not like, okay, somebody is randomly coming out of nowhere and, you know, kill me. No. It is also the protein itself who wants to kill itself or go or do the suicide, because he also wants or she also wants, I don't know the gender. They want that the cell should do what it's supposed to do. So they just do this sacrifice. They put this red flag, kill me, the other protein comes and kill it, and then that's how a cell divides. So this is actually, yeah, as you said, it is a team effort.
And you talked about willingness of sharing data, let's say it like this.
I mean,
how do you get data out of it if a thing, your meteorite, doesn't want to tell its story?
I mean, like they definitely, so meteorites, stars, planets, disks, they all definitely have stuff going on there, and like that stuff is visible and we can measure it. But it's never, we're never in control of, we cannot ask a question to a meteorite. We just get what we get, and the same with the protoplanetaries. Can you maybe form a planet right now, and we're gonna see how this band changes or something like this.
Mm-hmm.
We don't do this. We just get what we get.
Mm-hmm.
So I mean, they're willing to communicate, but they never listen to us what we ask them.
When I hear this, like we get what we get, Carlos, when I take you in on this one again, what do you do when you're stuck on stage? Because you also only get what you get. If your acting partner is not delivering an idea, a concept, a line that you can easily hook on, you might get stuck a little bit. What do you do? How do you resolve that issue?
Like Mamma Mia.
Then you basically stay on it. You stay with it, and you should be okay with that very real possibility of failure, and you have to just write it. I mean, you have to just. I mean, I was thinking. I think when you get a suggestion, you don't know what you're getting, right? But I can see that for cosmochemists, it's even more difficult. You cannot even ask for the suggestion, right? So we get it even easier than cosmochemists, and they still getting stuff out of the labs, right?
Yeah.
So we just staying with it and being okay with the possibility of failure, and we are all. I mean, our minds are really wonderful things, and we if we want to communicate, we want to get the scene somewhere, we will.
Mm-hmm. If there are any questions are popping up at your place, just raise your hand and I promise I will keep an eye out for this. Yes?
With the possibility of failure, there is like a beautiful essay written that to do science, you need to accept that you're stupid because you're never gonna know stuff. So you just have to accept. I mean, obviously you're not stupid if you in general people are not stupid, people are smart. But like if you do science, you need to accept that you're gonna feel stupid because whatever you're gonna do, you're gonna know nothing about it.
So that's this classic
And
conundrum. You find more questions.
There's an essay about importance of stupidity in science. It's like beautiful.
Interesting. So like this acceptance is one thing, but I want to put out another. Is there a question? Yes, there's a question, please.
So recently I read an article about how much humor you need in science, and then it's often the problem that humor is not a universal thing, and maybe that is also something maybe for theater, like perspective, how you can like, because on one hand it's good if something is a bit like you have a pun somewhere in your art, like for example the paper title or something, but then it's always the question like how can you be humorous or like maybe like less serious, but in a way that it's not like everybody can see, like understand it. Like when you do like theater, like how do you make sure that like you're not, well, like some people don't just like everybody kind of gets the joke.
And I think that's especially relevant for you because you did comedy or improv theater in Spain and America, now in Denmark. So how do you adapt to different styles from the audience?
That's really related to what I talk about context. You have to be aware of the context and you should read the audience. Like when you're doing so, for example, in Copenhagen, it's not the same if you have an older audience or a younger audience. I mean, there's a few things that we all relate. I mean, we all relate to vulnerability and like connections between people, and there's some, I mean, a lot of little things we can relate. And all of the, whenever you create something with the audience, it's like you have an inner, we have an inner joke today with like potato flowers. I mean, nobody else will get this. I mean, you can tell whoever you came with today. I mean, next time you see a flower, you can be, maybe it's a potato flower. You can laugh about it, and nobody else in the room will be like, what the hell is going on? So this context, we are creating context together. So there's a context that you have to be aware, and there's a context that you create in the performance. And that one, if you take care of that one, you will do something beautiful.
Like also like the whole context, you also need to adapt your performance to yourself, or not to yourself, to your audience. And that's a question maybe for you, Charu, and for you, Carlos. And that is, how important is this adaptation? Like you already mentioned a little bit, like you need to adapt to your co-actor, your audience, but you also spoke about that the proteins need to adapt to each other. And I remember doing some talks where you said like, yeah, they can both change the language and then it may be working again, there's no problem. So how important is adaptation for successful communication? Maybe you start on that.
Yeah, it's also context dependent. Sometimes protein just doesn't want to adapt because that's what they're not supposed to do that, right? But sometimes, okay, let me put it in a way that we have 30,000 genes and we have millions of proteins in our body, right? But not all the proteins are equally important, right? So sometimes the cells are okay if you don't have one of those proteins, but if you don't have the other protein, then the cells just die, right? So it's like the adaptation, how cell gets adapted, and it can just do regular functions even without having one particular protein because it doesn't play much role. So adaptation also comes how much important role a particular protein is playing in that cell, otherwise it will not adapt and it will just die. That's why in the video that I showed in my introduction, since the process that I'm stopping using that drug, cells are not good with it. Cells are not happy with that drug. They are not willing to adapt. They just die. They don't form those blue dots or the colonies. So adaptation is also context dependent, like which protein, which process you are stopping. If it is like you're stopping DNA damage, no, a cell cannot cope up with DNA damage, right? It has to be repaired. But if it is something like smaller thing, then there are other proteins that can, come up and help with the process. So cell is fine to adapt in those situations. So.
And I love that you just naturally, and I think we all do, we use like these expressions like this protein has a role to play,
yes.
Which gives me a beautiful bridge to go to the theater. This idea like there are some things, some roles you need to pay attention to, and there are some other roles that are not so important. Are there some details in your performance where it's like, okay, if this doesn't fit together, that's not important, but other things, they need to be spot on, they will need to have all this accuracy.
I think in general, just the trust of the partner and looking for making sense, right? I mean, you have to you cannot negate what the partner is giving you. So if Erika tells me this is a potato flower, I cannot reject that reality because we're building it together, right? I mean, if I reject that, then the whole date and situation we created in front of the audience, it disappears, right?
Mm-hmm.
So that's that's something they cannot go without. You cannot go without acceptance, and you cannot go without acknowledgement of what the partner is giving you. So this trust has to be there.
Is there a minimum of trust that you have to put into your partner or your data? And that's something where I mostly look at you because you're working with these trust issues, or like how confident are you? Or we call it confidence issues. What's the minimum amount where you do trust your data? Is there a threshold that we can look at?
I mean, usually every scientist, I guess, trusts its own data, and it's purely because we're human and we're biased to think that we did a good job. And also, it's completely, I was going to say natural, but it's not natural. It's completely necessary to never trust any data. It's just like you need to check it, and like repeat it again. So, and so what happens with at least plain information studies? It's always all of the data we get. It's like literally on the edge of like resolution. So with the questions we try, it's always on the edge of resolution. It's always we just barely caught the difference between two meteorites, or we just barely saw an exoplanet, or it's always.
So we could say it's like a 1 second movie. We just got the cornerstones.
Yeah,
yes. There was a question back there. Yes.
We're now talking about all these essays on science, right? And one of the big criticisms of the natural sciences is that instead of making new great inventions, people are getting more and more specific on what they're studying. Now we understand proteins even more. Now we understand this meteorite even more. So I was thinking for the two scientists, could you use improv as a way to maybe gain these new ideas or wild hunches that once dictated the natural sciences, right? Do you think you could go home to your labs and say, you know what, put the pipette away, let's do improv?
That would be wonderful. I would love to do that.
On how many things can you destroy the lab when you're improvising with your chemistry or with your biology?
With this hazardous substances, so you cannot really do a lot, right? So you don't want to mess up with something that just causes cancer right away. Like after 10 days, you're just on the bed, right? But I really like your suggestion that, or improv, like you should improvise. You should not just follow standard protocols that somebody in your lab has established 10 years ago, right? You should always be experimenting and trying different stuff out and see if you see something different. And trust me, we do this all the time and every day and all, like in every experiment. I do not follow protocol that was there in the databases. We tried, how does it look in my hands? If it looks in same as my senior said 10 years ago in my hands, otherwise I do not follow that protocol. So we do improvise, but it is only limited stuff that we can just experiment with when it comes to lab, because you know you're working with some harmful substances in the lab.
We want to do pandemic for you,
But you say like you improvise, you iterate on your recipes. You showed us in your video, okay, here's the recipe, how I extract nickel. It was nickel, right? Are you or your colleagues or your supervisor, somebody sometimes checking on this one, improvising on this one and trying to find a new solution?
I mean, yes, in general. So, but it was actually for me, particularly for me, it was vice versa. So with my one of the chemistries I did, it was in nickel chemistry, because I studied nickel and we really imaginative here. And so my supervisor just gave me a recipe and like, yeah, okay, I did it like 5 years ago, it worked, so just repeat it. And then I did it, didn't work. Then I tried, okay, maybe it was this thing. Then I did it again, it didn't work. And then like I basically spent the whole summer, like 3 months, trying to figure it out. And then okay, So my supervisor always told me, yeah, okay, like you can change stuff, but don't change this thing. And I changed this thing, and finally worked.
How do you, Carlos, avoid this trap that when you have a successful performance, like some lines that always work, they always are funny, people look up, that you're not using them?
So I think it's because it's not really the line. I mean, like the pony ride was great, but I mean, on its own, it's because of the world that we created, right? And you will not have that word next time you do a performance. So you cannot bring, the audience will not be the same. So they will give you new suggestions, and that's the beauty of it too, that then there is nothing that I will say in the stage that is like something I think, I am home and this is a great joke, I'll try to use it, because it will not work. Like the real good theater happens when there's vulnerability, there's trust, and there's something that happens in front of your eyes. And that is what is beautiful to see.
And I think on this sentence you said, they're like trust, beautiful things are happening when trust is there. It's a beautiful note to close on. Unless there are any questions left in the audience. There's one, yes, thank you.
I guess this question is a little bit more to the artist and less to the scientist. But when it comes to human communication, there's this problem. We have very short attention span. How do you deal with that? Like when you're on stage, do you worry that the people might just get lost in their thoughts and not sit and listen to their thoughts anymore? And I guess maybe for the scientist, when you're writing a paper, you think about it.
Maybe you start.
I would love to hear what you guys I think, so that's a great point. But I mean, if you go to see a show and you participate into the show, you're engaged to it. And I think, in a way, the stakes, I mean, the fact that we don't know where it's going, that is something that keeps me on the edge of my, to keep you in the edge of the chair, and it keeps me really awake and present in the stage. But it's something that danger of not knowing where it's going. I mean, danger. There's no life threatening here, but it's really not knowing where the show is going. That I think that it hooks the audience because you're seeing it happen in front of your eyes, right?
Yeah.
And I think that's where, if you miss it, it will never happen again. There's no way to review that. Like if you're watching a movie at home and you're like kind of sleepy and you just like kind of get more comfy in the sofa and close a little bit your eyes. Doesn't matter. You can watch it another day. You're tired today, so whatever. But you cannot miss a second in an improv show because it was there and it's gone and it will never be there ever again.
I want to mention that we do tape tonight, and if you find it really hilarious, you can see it again. But Charu, like maybe first for you, like this idea also like when you write the papers, like that can be really boring, you need to be attentive, and you have this delayed gratification, right? While you're writing, you don't get anything out of it, you get it a month later, half a year later, when somebody says like, yeah, what you did is good. How do you cope with this short attention span that might result out of this?
First of all, I'm a very early bird researcher. I just started my Ph.D, so I've never written any paper for that matter. But I have written articles, I have written like literature review when I have to submit my report, like how am I doing in my PhD. So I do get this block, and the only way out is stop writing, go to the lab, talk to people, and then I think it just helps you if you just get a break. You don't have to just sit and just keep writing. And I think you're not efficient if you're just sitting and forcing yourself to write something. You need to get a break, go and talk to people who are not from science, because if you're meeting people from science, they're going to talk about science, and you still are in the back of the mind, okay, I also need to write some report, right? So you need to just take a mental break, not the physical break from writing, and then I think it can help you.
Georgi, you have a couple of publications out. Do you?
Not really. one from way long ago.
Really?
Yeah.
The Crimea one?
Yeah.
Yeah. But do you agree like that when you're stuck, you need to take a mental break? You said it, or do you have some other tips maybe for Charu that is starting to write now?
No idea, because I'm right now struggling my way through my first actual, like where I'm the first stuff where I'm actually doing stuff. it's I haven't figured out my way through this yet. Like it takes me ages to do my first paper, and like right now I'm like revising it for the I think 12th time.
Oh, nice.
And it takes me months, so it's beautiful. Once I figured it out, I'll have some advice.
I can also say from my experience, like there's not a single scientist that enjoys writing paper. Everybody enjoys finding something new, doing experiments, trying something out, but then reporting on it is always a bit like, yeah, what did I do and why did I do it and why do I need to defend this?
Reporting on conferences is a completely different thing, because for reporting on conferences, you can skip so many details, and you can also like, you just need to get your stuff into 15 minutes and you're good.
Yeah.
But then it comes to the context thing and like trying to communicate with people, because usually people who listen to you, they didn't do this and they have no idea what you're doing.
Yeah.
And if you do stuff like me, it's not just geologists who didn't do exactly nickel. No, it's also astrophysicists who didn't do any geology. So you need to actually bridge a lot of people together to explain just 5 minutes of your research.
So because we already used much more time in this talk than I wanted to, maybe I can summarize what you also just said. Is like talking to each other and have like in both directions, the communication is nicer than have it in one direction only, where you are telling something to someone and then you don't get feedback. Then it gets a bit boring. Would that be right? So I think like feedback from other people is really important, which I think also resonates with you. And feedback from other proteins is also important so that we can divide our cells, right? Yes, thank you for this. Yes. That brings me to you. I really hope that you all enjoyed tonight. I hope that you maybe even learned something by accident tonight. That you feel a bit smarter, and that you have and you also got this idea that science does not have the answers, but it's our best way to get some new answers by checking with other people, with other data, with other scientists. I think we should thank our speakers for being here tonight, and also for using all this time to prepare the contributions tonight, and being brave enough to be on our third event here after 2 other great events that we had before. And I think you guys completely exceeded at this one. And I want to extend this thank you also to Erica, who's sitting there in the front row now, who was struggling her way through Mamma Mia and did great on the improvisation. Thank you, guys. And to replenish on your blood sugar, you get some chocolate, and maybe you can share a little bit with your partner there. Good. With this, I have to say this was our last BluBulB event for now. And I was hoping for some... Oh, thank you. Thank you. My colleagues Moreno, who's sitting over there, and Eleonora over here, we had a huge blast organizing these events, doing these events here, and we also hope that we can do some more events in the future. But for this, we need some funding. And we believe that bringing young scientists together on a stage like this and being cross-disciplinary, having experts from different areas, from different fields, and an artist in between really creates an environment that fosters ideas, where new ideas are born, and that really brings science, art, and this whole accessibility to science forward. But to do this, we also need you, because we need your feedback. I said this in the beginning, and if you stayed so long here with us, you really liked it, I hope. Use these QR codes that you have in front of you and let us know why are you here, who told you, did you like it, what can we do better? We also, though our whole project is a little bit for now, at the end, please follow, subscribe, like us on all the big social media channels, Instagram, Facebook, YouTube. Twitter, thank you. We're also on Twitter, because you really give us this leverage to make new things and to say what we are doing has some people that are interested in it. Well, with all of this, the 6 of us are here tonight a little bit longer. We are open to talk, to chat. I think all our contributors, if there were any questions you either haven't asked yet or didn't to ask in the plenum, approach them. They are happy to answer them. I answer on their behalf. We are here a little bit longer from organizing this one. Thank you, everybody, for tonight. Thank you for coming.